All right, so this is week 4 lecture of the neural basis of consciousness, and it's an introduction to the visual system. And we'll start from the recap of week 3. We first started with studies of the brain lesion patients and discussed what consciousness is and it is not, and it can be from those patients' phenomenology and observations. And the phenomenology brain linked data is very important kind of standard that any theory of consciousness need to explain. And secondly, locked-in cerebral agenesis damages in the prefrontal cortex patient has been also discussed. And here, the important thing is that the conscious experience of a patient can dissociate from externally observable behaviors. So in the case of locked-in syndrome patient, they cannot move in terms of body. They can only blink and make an eye movements, and still the patient that I described was able to even write a novel. And then the cerebellar agenesis patient was born without cerebellum, but was able to function as a mother even in the end. And then damages in the prefrontal cortex typically changes social functioning, but doesn't seem to change the conscious experience. So architecture of the brain area matters, not only the number of neurons, and that's most clearly sort of reflected in which areas of the brain. Do you know? Can you tell which among 3 types of the damage, which one you think is the most relevant for this conclusion?
I don't remember, sorry.
It's a cerebellum. Okay, so cerebellum contains roughly 4 times more neurons than the cerebral cortex, entire brain, and that's very important kind of fact, and you should remember. Okay. All right, and then the executive social functions can be impaired with intact conscious seeing, hearing, and feeling. That's what I said right now. All right. Then other important fact that we covered the last week was the substantial portion of brains can be damaged, while apparently keeping a hydrocephalus and hemispherectomized patient functioning, and they are conscious. I don't know, do you guys remember what types of the patients they were, hydrocephalus and the hemispherectomized patients? Do you remember what these terms mean? Okay, so just to recap, a hydrocephalus is the one that had water in the brain, and then that put a lot of pressure onto the brain tissues, so that all the small the brain tissues are kind of marginalized, and the very small island of the cortex only remains. And hemispherectomized patients are the ones that are cut in one of the hemisphere and had only one hemisphere only, but they were quite well functioning and conscious. Okay. But by the way, can you hear me? Fine?
Yep, yeah.
Okay. All right. Yeah, it's not locked in patients. Okay, let's go. And then after losing posterior visual area, posterior means the back of the brain, okay? If these parts are lesioned, then a patient reports loss of visual consciousness. So the visual aspect of the consciousness is highly, sort of the strongly supported by the back of the brain. And if you lose that parts, then you lose also visual consciousness. But the blind side patient and monkeys are the ones who have lesions in the back of the brain, and then also reports or behave as if the visual consciousness is not there. And if you ask blind-sighted patients, they say that, no, I don't see anything, okay? However, when they are forced to do that, then they can show some kind of performance based on the visual ability. Do you guys remember what kind of evidence you saw in blind-sighted patients or monkeys? Either of the movies. The man avoiding obstacles. Yes, yeah, that's the one. Okay, so that he wouldn't walk spontaneously in the room, but when he was forced to, he was able to move around. That's right, exactly. Great. All right, so this type of lesion patients evidence left us a lot of questions. And first of all, question is what it feels like to be like them. And another one is what kind of visual inputs to the brain are there. And then that leads to these types of non-conscious visual behavior. So learning objectives for week 4 are the following. One is that what are the properties of visual inputs to the brain? So here I'm separating the inputs to the eye to the inputs to the brain. Okay, what the brain receives is, as I showed, relevant for consciousness, and the property of the visual inputs to the brain is different, slightly different from the inputs to the eye. And then secondly, how does the structure of our eyes shape our conscious vision? So our eyes are structured in a particular way, and that changes or modifies the way we see the world. Okay. And then the third one is that what is our peripheral visual experience like? And how the theory says about it. And then fourth, how can we explain the properties of visual consciousness based on the architecture and the mechanism in the visual system? So this starts to become like a link between how we see it, consciousness, and how it's supported in the visual system. And this is a reading materials. It's not necessary, but I suggest you read it. And where quotes are coming from is only 5 pages for chapter 3. So that's it. So so far, any question about contents last week? Fine. Okay. So from this week, there will be somewhat, controversial kind of topic that you will hear. So this is one cartoon about sort of the way people see the world, and also actual world is, whereas something that you might actually learn in the university. So it's a kind of meta-scientific kind of discussion. So many cases, your lecture so far, up until like 3rd year or 2nd year, you tend to receive some kind of a story about the world or how the mind works in psychology in a way that is very simple and understandable. And that's, for example, it's a really good book, but you also need to be careful about this. But this is Daniel Kahneman, who is a Nobel Prize winner, proposing that our mind works in a fast component and a slow component. And the fast component is like a non-conscious, unconscious, emotional, animal-like. And the slow one is a conscious, rational, and logical kind of thing. And if you read this book, which is actually quite long, so it may be difficult to read it through, but I strongly recommend if you really want to understand sort of how many people work. In fact, this book itself challenges many of our intuition, and it has lots of useful kind of a reality that says that reality is actually not that simple, but it's actually much more complex. and then complex but correct. So this complex but correct kind of pathway is a winding road like this, and it's difficult one. So not so many people can take it, tolerate it. But the simple but long is the one that is much more popular. And one of the bigger long-term learning objective of this class is that to open up your eyes into more on this side. Okay, so I start to inject some of the topic that many people believe is true, but it may be not true, and especially in relation to our conscious experience. And for that purpose, I strongly suggest that this book called The Think Again by Adam Grant, and it's sort of the way I want you to think about what you have learned in university. So many of the theme of this book is that as the world is getting faster and faster in generating new knowledge, many knowledge which was taught in the past or believed in the past start to become unstable, and also what is necessary for us is to unlearn what you have learned before and then adapt to the new things. And for that, it's quite important to take this kind of mindset. The reality is quite complex, but there's a way to understand it. And to get there, you might be able to start with a simple kind of model or thinking, but eventually you need to face with the fact that reality is quite complex. And that's something that as a psychologist or even a practitioner of psychologist, or even if you work in industry, it is quite an important kind of lesson and also useful lesson to learn. So I strongly suggest this book. And if you read these two books, you probably will learn much more than any combination of all the lectures in the class that is taught in probably Monash psychology, I think. Anyway, so so far is it fine? Does it make sense? I mean, it might not make sense at this stage, but I'll try to explain what I mean by that today. So let's start. What is the properties of visual input information to the brain? Do you see the blueness at the place where you're looking at? So that's a kind of enigmatic kind of title. So let's move on. So the properties of the visual inputs to the brain are dependent on the structure of the eyes, and phenomenology, our conscious seeing, is shaped by the properties and the structure of the eyes. And important structures include that retina, that is the back of the eye, that has lots of photoreceptors, that is the basis for the visual consciousness. And then optic nerve that sends the input into the brain. And then within the retina, photoreceptors, cones and rods. Cones are responsible for the color, and the rods are not. And then retinal ganglion cells, and fovea is the area that you are looking at directly at the fixation. So this is a gross anatomy of the eyes. It's consisted of various components, lens and optic nerves, and so on. So I'll just skip these anatomical details. And then this is the electron microscopy of the slice of the retina. So in fact, the world is located around here, and then your eyes process or look at this world like this way, and then projects in this way. And the retina is a two-dimensional sheets that's surrounded around here, at the back of the lens. And then if you look at the cut slice in a two-dimensional way, Then that's what you see here. Okay. So one thing, does anybody know which direction is the, I mean, up and down? Or maybe let's ask the quiz. Okay. So option A is that in this figure, right is coming from the top. And then option B is right is coming from the bottom. So because I haven't actually explained or told you, it's highly probable that you don't know anything about this, but just by hunch, just tell me which one is what. By the way, this is the photoreceptors. So if you see these things, as I will explain, these are the photoreceptors, okay? So more or less, you all responded? Okay, good, everybody responded. So the correct answer is this, actually. Here, this is the optic nerves, meaning that it is the axons that sends an input output from the retina to the brain. I mean, to the lateral geniculate nucleus in thalamus, to be accurate. And in this way, lights are coming into the eye, and then photons are caught by these type of structures. It's very weird. It's very weird why we have this kind of structure, but this is how our eyes are constructed. Okay. So if you know anything about optics or how the light works, this means that any kind of photons that goes from here may be scattered by this types of difference in the membrane, or maybe sort of the diffracted by different angles, or sometimes absorbed in this kind of intermediate structure. So it's definitely not an optimum structure to see world accurately. That's the most important thing. Okay, the eyes are not constructed to be the optimum, highest resolution as possible from the mechanical or engineering point of view. We usually don't notice that, but that's how it's been there. Okay, and this is as I said, this part of the retina is the rod and cones photoreceptors. And if you look at the individual rods and cones, it looks like this. So it's like this part that contains lots of photoreceptors, photosensitive. So the segment is the tip of rod, and then cones have this, and then shapes are these shapes are the origin of this name rod and the cones. And so I flipped the image here so that you'll see that eyes are receiving the light from this way, and then catch the photons around here with these cones and rods. As I said in the last slide, so it's not optimum to see this kind of to process these optical input. And here's the biological kind of proof that eyes doesn't have to be constructed in this way. So octopus, octopus, right? Living creature in the sea, they have in fact the structure that has the rods and the cones up front. So there is no scattering of the lights with it because of the lots of structures up before the cones and the rods. So they they are, in a sense, constructing the eyes much more, logically and a very nice, nice way. But no vertebrates, they have all the retinal structure. It's in an opposite way. Do you know? Does anybody know that there is also weird kind of fact that about the eye structure in the mammalian or vertebrates eyes? Something that is not super straightforward if you think about how the words are projected to some kind of visual pigments. It's a bit difficult to probably say, but yeah. So the other weird thing is that it is upside down. As I depicted in here, the light comes from up, and then the image on the retina goes in a sort of the opposite direction. So something that is projected in here, let's say there is a circle with respect to your fixation to the left, then that's projected onto the right side of the eye. And so the bottom, things on the bottom gets projected the upper part of the retina. And the octopus doesn't have that kind of inversion as well. And does anybody know who discovered this kind of weird architecture of the eye in the history? It's quite striking that I also didn't know until really recently, but it is yeah, it's written in the Bar's 2009 book, in the one that I suggested as a suggested reading. And here I'll tell you the answer. It's an Descartes. And I think I'll check later, but I think it was Descartes, and he discovered both inversion of the eye and also the aberrant structure of the eyes as well. What is the consequence of the deflection in the vertebrate retina? We are worse at hand-eye coordination than the octopus. That's an excellent question, actually. The answer is that I would say it's unclear compared to the octopus. I don't know, but the fact is that our visual system is extremely good at doing many things. And for example, even though there's a diffraction and reflection, the probability that you can detect one single photon by the eye is very high, actually, even in human eyes. So it's almost operating at the optimum situation, but it also contains many different kind of mechanism, including adaptation and also calibration of the visual system and things like that. So at the level of the retina, it looks like octopus may be better than the vertebrates, but it's not really clear as the visual processing goes, whether this advantage is preserved in the octopus. Okay. So then the quite important structure is called fovea. So this is a part of the central issue today. This, dipped kind of structure in the eye, which corresponds to where you are looking at, has the highest resolution in your vision. And this one is just about 1 degree of visual angle. I'll explain what it means. And it's a width of your thumbs at arm's length. And it's slightly narrower than that. And it specializes process the vision at the highest resolution. So I'll come back. Now it's 25 minutes. So I'll take a break, a 5-minute break, and then start from here. Okay.